Yes, hello everybody. It is my great pleasure and honor to be with you again. And this time it is about application of natural products as a pioneer natural sources for green synthesis of nanoparticles, nanocomposites, and their potential application. I would like to take this opportunity to sincerely thank collaborators in China, where I am now, for great hospitality. Let us talk about nanoparticles in a very simple way. Short introduction, one example of our experimental results, and then application to marine. Regarding nanoparticles, it is a particle that exists in nanometer scale, and it is usually between 10 nanometer to 100 nanometer. Its properties, uniformity, good electric conductivity, stability, catalytic performance, magnetic property, and biological activities. What about the types of nanomaterials? It can be zero-dimensional, like nanoparticles and quantum dots, one-dimensional, like nanowires, nanotubes, nanorods, two-dimensional, like thin nanolayers, nanofilms, nanosheet, three-dimensional like nanocomposite. What about methods of preparing? It can be top-down, cut object smaller and smaller until attain size needed, or bottom-up, add atoms together one by one to attain correct. So this is a way top-down, and this is a way bottom-up for nanoscale structures. Applications is very wide, from textile industry, water treatment, agriculture, food industry, drug delivery. Then it comes to the idea of the senses. It has been somehow as a replacement, you can say, of synthetic procedure, which developed first by forming stabilizing agent for silver nanoparticles. So the biosynthetic metallic nanoparticles can be prepared using plants, algae, fungi, so different extracts of natural products. In this case, it has the secondary metabolites of different organisms. It usually include flavonoids, alkaloids, terpenoids, phenolics, enzymes. And green senses of metallic nanoparticles using extracts as a senses stabilizing and decapping agents. If we compare it, the green senses advantages is easily available, comparing it to the other which contain more costly equipment. It's also cost effective regarding expensive way, and more also it is eco-friendly comparing to the usual synthetic methods regarding environment. We take one example about Chile and see what's the formation when we add titanium oxide and also silver nitrate. We have a very good experience about chemical profiling for characterization of the chemical constituents using LC-MS, and NMR for identifying several classes of the chili, as you can see. The method for preparing is very easy. So we take the fresh chili and then stir it with water for 2 hours. Then add the silver nitrate, heat it for 2 hours at 70 degrees to form silver nanoparticle. We follow that by adding titanium oxide and stir it for about 1 hour. Then after 3 hours, then centrifuge and drying to form chili composite. So it is very easy method and also eco-friendly. Then it comes as a difficult part for characterization. It can be photocatalytic evaluation, like using a reagent for coloring reactions, like using, for example, methylene blue and stirring for about 60 minutes. It can be also by using dinitro aniline. Then it comes the results, and the most important is the morphology, structure characterization, and photocatalytic studies. So if we use high resolution transmission electron microscope, this is very efficient method to see how the size, if, for example, here 10 nanometer and here is 5 nanometer. And SEM, and also we use also X-ray, XRD, and this you can see here how it look like for the nanoparticles. The infrared, as we all know, that it characterizes the functional group like hydroxy, amino group, etc. Then XRD, this is very efficient method. And here, as you can see for silver and Ag2O and B25, as illustrated in the chart. Also, TGA is a very effective method. And here you can see for Chile and for silver nanoparticles and for the composite. So the Chile extract started to compose into the volatile and organic compounds, as you can see here. And the blue color is silver and the other blue is the composite. And photocatalytic evaluation study, this, we can study this using the equations shown below. And accordingly, we can see the time course degradation curves and the linear plot of the composite. So here is 3 milligram and then 5 milligram and 10 milligram in red color. And here at different times, starting from 10 to 60 minutes. So you can see the illustration. And this is what we get the conclusion for this study. The degradation effectively was calculated to be about 98% for 3 milligram at 35 minutes, 62% at 5 minutes, for 5 milligram at 60 minutes, and 76% for 10 milligram at 60 minutes, respectively. And also the greater availability of specific surface area and pore volume increased, as photocatalytic performance is a surface phenomenon dependent. And also at the low dose, the notable decrease in the turbidity of the working solution that scatters light domain and maximize the photooxidation activity to stimulate the catalytic surface rather than higher catalytic dosage. Then here also you can see here at the different times, and also the NADH can catalyze the suspension in contact with 2,4-DNA. This is what conclusion we have of the photo reduction by green heterostructure of the composite. So, and this is a simple figure to show the photocatalytic degradation mechanism of MEP and the reduction of 2,4-DNA using the biosynthesized composite. So you can see the nanoparticles when it is formed, and then adding of titanium oxide as shown in the figure. What about the advantage of green biosynthesis of the composite from previous literature data? It is simple, non-toxic, and environmentally. Chile extract contain wide range of metabolites, including alkaloids and phenolics. The green synthesis of nanoparticles eliminate the need for stabilizing and capping agent and show shape and size dependent activity. The spicy test and the sharp order of the Chile was desperate after nanoparticles formation. So the smell, of course, it is different. And the choice could support the chili nanoparticles formation or different application, especially for the people who don't prefer the spicy taste. This is our conclusion. We go to a different application of marine organisms as pioneer natural sources for polysaccharides, protein for green senses of nanoparticles and their potential application. Here, the picture shows you like graphical abstract for the green senses of nanoparticles and what method to use for characterization, TEM, SEM, and what is the application in drug delivery as antimicrobial, bioimaging and biosensor, nanofertilizer, and photocatalytic. We can make it in very simple figure for the marine organism. When it reacted with metal ion sources and by reduction, then we get the metal nanoparticles, and then stabilization to get capping agent for enzyme functional groups, and finally the capped stable nanoparticle. So this schematic diagram of the proposed mechanism of the green senses via sulfur nitrate. What about the different marine sources? As you can see here, it can be different types of algae. And here also different microbes. And here also is different animals, so including shrimp, of course, spongy as well. And here is the proposed antimicrobial mechanism for nanoparticles as application of marine organisms mediated nanoparticle. So it binds with protein tissues that lead to distortion of the bacteria wall and molecular membrane. This could bind to DNA and RNA, of course, via denaturation, cause inhibition, bacterial replication. And it can interfere with the surface containing biomolecules in the bacterial cell membrane, or can strike the genome and respiratory chain, and ultimately resulting in the tips of the bacteria cells. We also studied the proposed mechanism for anti-cancer activity and drug delivery, and also, as you know, anti-cancer related to antioxidant, as you can see from the proposed mechanism of nanoparticles. Also, we studied the proposed mechanism for degradation and reduction of dyes utilizing silver nanoparticles, as you can see from the figure here. So this is our ongoing project also about recent insights in the potential application of nanoparticles by marine organisms. And we come to this conclusion that the current innovation in biotechnology area are supporting the stimulating developments other fields, including nanomedicine, pharmaceuticals, sensors, environmental trends, food and agricultural aspects. Natural product can biogenerate a broad spectrum of phytochemicals, metabolites, including proteins, peptides, alkaloids, flavonoids, carbohydrates, polysaccharides, etc. as secondary metabolites. These products exhibit exclusive advantages that offer pioneering roles in the eco-friendly fabrication of several nanoparticles with different metal oxides within a single phase. These nanoparticles and nanocomposites show several medical applications as anti-cancer in drug delivery and antimicrobial. This is our target. This is some of our publications in this area. I would like to take the opportunity to thank very much all the group for excellent contribution. I wish you all the very best, and please kindly give us your constructive criticism to be better all the time. We must work on ourselves. Thank you very much for your kind attention. See you again.